It is an honor and privilege to receive the scholarship named after Dr. Grosfeld. You can see here in this picture that I took at the BAPS meeting in Dublin in 2005. Whenever I updated him about my career progress, it was always very encouraging, and I remember I had a chat here with him in San Diego at APSA 2016 when I had the chance to introduce him to my then 3-month-old daughter. Thank you, Dr. Grosfeld, for being such a source of inspiration for us all. It is well established that pulmonary hyperplasia plays a central role in the pathophysiology of CDH. It is one of the main determinants of clinical outcome for these babies. In broad terms, we can say that pulmonary hyperplasia is characterized by impaired lung growth, maturation, and vascularization, whereby CDH hypoplastic lungs have fewer branches and air spaces with undifferentiated epithelium and mesenchyme, and fewer vessels with increased medial wall thickness. There is a consensus that the prenatal period offers us a window of opportunity to promote normal lung development, and an ideal therapy would be one that could address all aspects of pulmonary hyperplasia. So in search for a regenerative therapy that could promote lung development, my lab has been studying extracellular vesicles derived from amniotic fluid stem cells. These are nanoparticles that, when released from stem cells, they are the key mediators of their paracrine signaling. We reported our optimal strategy for isolation using ultracentrifugation and the EV characterization according to the guidelines of the International Society for Extracellular Vesicles. Last year, we reported that administration of FSC-EVs to several models of pulmonary hyperplasia rescues branching morphogenesis and promotes epithelial cell and fibroblast differentiation. In a more recent paper that is under second revision, we investigated the EV effects on cell differentiation at translationally relevant time points. And in this study that is about to come out in the Blue Journal, we highlighted the EV rescue effect on impaired autophagy signaling in hypoplastic lungs. When we looked into the mechanism of action of EVs, we realized that their effects are mediated by transfer of their RNA cargo, and we identified several microRNAs that are responsible for their regenerative effects. So with these studies, we robustly looked at the ability of FSC-EV treatment on promoting lung growth and maturation. But what about vascularization? We know that fetal can increase lung growth in selective fetuses, but it does not rescue abnormal vascular development. And in this paper that we recently published, we highlighted how alternative strategies to reverse pulmonary artery changes are needed. And as attempts to reverse lung vascular remodeling have not been successful, we decided to focus more on the EV effects on the fetal lung vasculature. And this is the time when we applied for the funding to the APSA Foundation, and I'm proud to report today that thanks to the support of the foundation, we were able to demonstrate that FSC-EV treated rat lungs had restored lung vascular density, decreased median wall thickness, and restored expression of markers that are key for vascular development. To investigate cell-specific effects of FSC-EV administration to fetal hypoplastic lungs, we conducted single-nucleus RNA sequencing, and we were able to identify all cell types that are present in the fetal lung that are immune, epithelial, endothelial, mesenchymal cells. And this U-map, you see each dot represents a nucleus. And we found striking differences between CDH-untreated lungs and control, whereby CDH-treated lungs had different presence and distribution of clusters that were reversed back to normal by the administration of FSC-EVs. Details of our sequencing experiments will be presented at fetal diagnosis and treatment session on Saturday morning, so don't miss out. When we looked at the endothelial cells in isolation, CDH-untreated lungs had, again, significantly different distribution and gene expression compared to control lungs, which had an expression that was similar to that of lungs that had received amniotic fluid stem cell extracellular vesicles. Gene set enrichment analysis showed that CDH-untreated lungs have downregulated genes that are important for angiogenesis and vasculogenesis, whereas CDH FSCV-treated lungs have upregulated genes that are important for vasculature development. When we looked into the RNA cargo of the EVs, we identified several microRNAs that are important regulators of angiogenesis, proliferation, and migration of vascular endothelial cells. But are these effects relevant to human fetal lungs? Well, in collaboration with an industry partner, we were able to isolate and characterize the EVs from GMP-grade human amniotic fluid stem cells. We tested them on our established human pulmonary hypoplasia model that we presented last year at APSA. And indeed, we could replicate what we had seen in rodents. So FSCV-treated explants had restored vascular density, medial wall thickness, and expression of angiogenesis markers. So encouraged by these exciting results in rodent and human models, we're now about to test GMP human FSCVs as an additional therapy to fetal in a larger animal model in collaboration with Dr. DePrest. So I hope I've convinced you that FSCV do not only rescue branchial morphogenesis and promote lung maturation, but they also attenuate fetal lung vascular remodeling. And their effects are mediated by their RNA cargo. So we believe that administration of FSCVs represents a promising cell-free therapy for babies with hypoplastic lungs secondary to CDH. I would like to acknowledge the hard work of all the people in the lab, the help of our collaborators, and of course, the support of the APSA Foundation. Thank you.
This is exciting work. I have a question about the timing. When do you deliver the EVs, and do you have to redose them? And when you take this into human trials, hopefully, at what gestational age do you think would be the prime time for this?
Thank you very much for the question, which is very relevant. EVs work more or less like a pharmaceutical drug that most likely need to be given multiple times. And I have a slide. So at this stage, we're thinking of, as I said, collaborating with fetal experts and adding it to FSCVs potentially in the future. But the real vision is that we could have nanoparticles that are targeting the fetal lung and that can be given to mom, IV, and so they can be given multiple times. We've been giving them at the end of the gestation, intraamniotically, in a way that feeding, breathing movements are already in place, and the pups can actually ingest, and we've done those curve experiments to make sure that we had the EVs in the lung and reproducing the effects that we had seen in the explant models.
And are there any off-target effects?
That's another very interesting question, not that we know of, and this is the beauty of cell-free therapy. However, during the pandemic, there was a lot of work done by several centers looking into EVs as a therapy for ARDS, and they discovered that, for instance, they can induce a hypercoagulability status. So, of course, we're not there to translate this yet, but we're very hopeful.
Thank you.
Thank you.
Thank you.